Hi, my name is Mitchell Klump. I am an application scientist in the Research and Development Department at Fort Collins, Colorado. Today, I will be talking about sorting large particles at low pressure on the Invitrogen Bigfoot spectral cell sorter using the 200 micrometer nozzle tip. Sorting delicate, fragile, or large cells on a larger nozzle tip at lower pressure can confer many benefits. One of the biggest benefits is a reduction in shear stress forces that these cells may encounter prior to being sorted. As you can see here on the right, there are many different cells that vary in size drastically. Some small cells, those being T cells or lymphocytes, as noted here by the cancerous T cell line, the Jurkat cells, are between 9 and 18 micrometers in size, contrasted with one of the largest yet rarest cells in the body, the megakaryocyte, which has a size of 50 to 100 micrometers in size. If both of these cells were to be sorted in a sample on, say, a 100 micrometer nozzle tip, there would be many issues with that sort. For example, the nozzle tip could become clogged due to the sheer size of the megakaryocytes, or even if they were able to be sorted, you might have some issues with cell clumping, you might have some cell aggregation, or you might have some fragmentation of cells, which will lead to downstream problems, either being viability, potentially, or your potential efficiency with other cells of interest, including these small cells like T cells. Therefore, sorting on such a large nozzle tip in both size but yet low pressure can reduce a lot of those shear stress forces. This in turn can help enhance the cell viability of these cells post-sort, and this preserves the long-term survival of such large cells. This also reduces the likelihood of cell aggregation while sorting with such a high nozzle tip, therefore allowing for smoother cell sample flow and maintaining your expected sort efficiency. Currently, the Bigfoot has a 70, 100, 120, and 150 micrometer nozzle tip. You can see that as the size of the nozzle tip increases, the pressure decreases. This holds true for the 200 micrometer nozzle tip, which is operated at around 6 psi. Even though the droplets are quite large, as you can see compared to the other nozzle tips, the droplet geometry remains the same. And this holds true as well for drop delay, which has two distinct peaks, both forward and backward, and gives you great confidence for your sorting needs. Using the 200 micrometer nozzle tip, you can sort a variety of cells into different positions, those being left 2, left 1, or right 1, and right 2. Because there are such large cells that can be used using this 200 micrometer nozzle tip, and megakaryocytes are a very rare cell in human blood, we use the megakaryoblast cell line, MEG1, as a proxy for these cells and sorted them and showed good cell viability, pre-sort and post-sort, as well as show that they came to confluence after 7 days in cell culture. To sort these cells in one-way sorts, MEG1 cells were cultured in rPMI, supplemented with 10% serum, and an antibiotic cocktail. Cells were then resuspended in 1x PBS at a concentration of 1 million cells per milliliter and run on the Bigfoot at about 1,000 events per second. Using Cytox Advanced as a live dead stain, it was added both pre-sort, so we could sort live cells, as well as post-sort to assess post-sort viability. 50,000 cells were sorted into positions left 2, left 1, right 1, and right 2. The efficiency from each sort position, left 2 all the way to right 2, was just as good, if not better, than the efficiency that we would have estimated when factoring in the nozzle tip size, the frequency, the sort speed, as well as the drop spacing. The viability immediately post-sort of the MEG1 cells was just as good, if not better, than the unsorted controls. While gated, viability was around 100% for all cells in all sort positions, ungated viability was just as good, if not better, than the unsorted control, and was greater than 90% in every instance. However, post-sort viability does not mean that these cells survived long-term. To assess long-term survivability of these cells, the sorted megakaryoblast were put in culture, that being rPMI supplemented with 10% serum and an antibiotic cocktail, onto a 12-volt plate, as well as with unsorted controls. They were imaged daily using an EVOS M7000 imaging system. And you can see that by day 4, the megakaryoblast had started to grow, and by day 7, along with the unsorted control, cells were confluent and the media had changed color, indicating that they had proliferated. Using the Bigfoot, you can also sort megakaryoblasts 4 ways into the sort positions mentioned before, being left 2, left 1, right 1, and right 2. Megakaryoblasts were cultured, as mentioned earlier, in rPMI, supplemented with 10% serum, and an antibiotic cocktail. Cells were then resuspended at a concentration of 1 million cells per milliliter in 1x PDS, and sorted 4 ways. You can see in the bottom right histogram, using Cytox Advanced as the live dead stain, that each population was sorted live. The efficiency for each sort position during this 4-way sort was just as good, if not better, than the estimated efficiency given the metrics during the sort. The viability for each sort position was also almost 100%, including the unsorted control, when gated. Ungated, the viability was just as good, if not better, than the unsorted control. Proliferation of these cells sorted 4 ways was also very good, in that by day 4, the megakaryoblast had begun to grow, and by day 7, along with the unsorted control, in their normal culture medium, being rPMI, serum, and an antibiotic cocktail, the cells had grown to confluency and the media had turned yellow, as imaged with an EVOS M7000 imaging system. There are many instances in which case you don't want to sort a simple homogeneous cell culture population or sample, and therefore it's nice to have a mixed sample to test. Megakaryoblasts were mixed with cryopreserved peripheral blood mononuclear cells, or PBMCs, and the metrics of being viability, purity, recovery, and efficiency were analyzed. Here you can see a scatter plot showing our lymphocytes in orange, our monocytes in green, and our megakaryoblasts in blue, showing that they differ, how they separate from one another via scatter and how you can resolve each population. They were also stained with a variety of fluors and markers to sort from. Viability was assessed using a live dead stain, being Cytox Advanced, both pre and post-sort. Purity was assessed by reanalyzing the sample immediately post-sort and determining how much of each sorted population fell back within its sorted gates. This oftentimes requires a slight gate adjustment. Recovery was assessed by manual counting via hemocytometer, and efficiency was compared to the theoretical maximum efficiency for each sort. You can see here that both purity and viability were greater than 90% for our megakaryoblasts when reanalyzed post-sort. Recovery was also 100%, in that we sorted as many cells as we had hoped to sort. The efficiency for the megakaryoblast was also within a good range relative to our estimated efficiency when sorting such drastic populations. Thank you very much.